Good day, everyone. This will be our first activity in the chemistry laboratory, whether it's going to be for biochemistry or chemistry for engineers. It is entitled Common Laboratory Equipment and Apparatus. And in this activity, we will identify the different lab apparatus and equipment, as well as recognize the function of each, how these will be used in our experiments throughout the semester. There are about 50 to 51, but then they will not be used altogether. So it will vary how they will be used in each experiment. So the first one is acid burette. So it is calibrated. It is used to precisely measure and dispense liquids specifically for acids. So based on the illustration, it's either the glass burette or the PTFE. So this is actually a long apparatus and it is fragile, it is expensive, so it is specifically designed for acids. So if there are other reagents being placed, then this valve here will have a blockage. The second apparatus is the adapter. So the adapter is used to link the condenser to our receiving flask during distillation, which we'll figure out later on when we have the experiment on purifying compounds. So this is made of glass and it varies in size depending on the size of the condenser as well. So the water from the condenser or the condensate will pass inside, and this tip here is connected to the receiving flask. The third one is the alcohol lamp. So the alcohol lamp is basically a generator of an exposed flame. So the fuel inside is alcohol based on the name. So this is the wick, and then in turning off the alcohol lamp, we do not blow. Instead, we use its cover to avoid the spreading of the flame. So it is also known as an alcohol burner or a spirit lamp. Next, we have an alkali or base burette. So earlier, we are shown the acid burette. This time, it's for the base. So to differentiate the two, you look at the stop cock or the stop valve. So for base, the material here is a rubber tubing, while for acid, it's either the glass or the PTFE. Okay, so just like the acid, so this burette is designed for bases. So we use place base reagents here. Otherwise, blockage again. Then this is also expensive. Then we are told to be careful not to break or to chip off this tip here, because it will be charged to you if you do so, and it is also harmful if it can injure anyone in contact with the chipped edge. So next is the aspirator. So the aspirator is a bulb. It is also known as a pipetter. So it is a rubber bulb which is connected to your pipette, as the name suggests, and it is usually used when together with the pipette. So next we have the beaker. So it is a cylindrical container. It is made of borosilicate glass, usually Pyrex, and it is used for storage of liquids or mixing. Although there are calibration marks, we do not use the beaker for measurement of liquids in precise manner because it is not used for that. We have the graduated cylinder for that function. So beakers come in different sizes depending on the need for the experiment. Then next we have the Bunsen burner. So our Bunsen burner is the laboratory apparatus which is used for heating. So burners are really shiny when new, but over time, when the burner is frequently used, so it leads to discoloration. So expect that once you start your experiments, you will encounter an actual burner like this. So the color is already dark, and perhaps there are markings at the side. So this burner will be connected to the gas cock where the source of our fuel is connected, and we will have that more elaborated on the experiment 1, part 1. Next is the Buet clamp. So this is a Buet clamp, to which others refer to as a butterfly clamp. So it is used to hold the burette, shown on the right side of this presentation. So there are two jaws, so each burette clamp can hold two burettes. So normally for titration, you can hold acid and base burettes. Then next is the centrifuge. So this is a tabletop centrifuge, and it is used to separate liquids based on their densities. So if you have an emulsion, then you're going to place it inside the centrifuge, and the force will separate the mixture based on their densities. So inside, you will see that there are holders for test tubes or whatever container that would fit in here. So in using the centrifuge, this should be balanced. So if in case the number of test tubes do not reach or do not suffice, then you will be using a blank solution. So it's basically just a test tube with just water to replace, so that the weight of this centrifuge will be balanced. Otherwise, if it is not balanced and you turn it on, then charge again because the test tubes might break. Next is your clay flame shield here in the left. So it is made of clay, and it is used to resist heat and be a stable platform to hold other equipment such as your beaker and your crucibles while it is being heated over a Bunsen burner. So it is used to deflect the air. So our lab is windy because the windows are supposed to be open while conducting experiment. So this to avoid also spreading of the flame from the burner, to protect the people and the things surrounding the setup. Then next we have the clay triangle. So it is also used together with the clay flame shield, and the clay triangle provides a stable platform. So in some experiments in engineering, that will be for heat of combustion. So you will put your aluminum can on top of this triangle, and this triangle will be placed here. If you see these markings or edges, this is meant for the clay triangle. Next, we have your condenser. So this is a laboratory condenser. So normally you are going to connect these to the distilling flask, and on the other side this will be connected. So bali ni siya. The distilling flask will be connected to this side, and the adapter will be connected to this side. So your vapor will pass through this tube or channel here, while water will be supplied through this connected to the water supply through a rubber tubing, and then it will exit here so that your vapor will condense during distillation. Next, we have your crucible. So your crucible is ceramic. It could also be metallic, but in the lab we only have ceramic. So your crucible is used to contain or to place your substances when you heat them. For example, you place them inside an oven or a furnace where it will be exposed to very high temperatures, because the crucible will not be melted or will not be damaged since it is resistant to the high temperature of your oven or your furnace. Then we have your crucible tongs. So your crucible tongs are or tool. It is a tool with jaws that will be used to handle hot crucible and other hot lab glassware especially in heating. So when you start experiments with heating, so you use these to hold your evaporating dish, your beakers, so as long as it could fit your jaws of the crucible tongs. Next, we have the Desicator. So it is a glass container which is sealed, and it is used to contain or to preserve the moisture sensitive substances. So it is used to maintain low humidity, and it is used to dry samples. So if you notice, there is material below. So these small solids are actually silica gel. Usually it's silica gel, and it is called your desiccant. So silica gel may be familiar if you open your newly bought shoes, for example. So you will find that there. It is used to maintain low humidity within your container. That's why you use the silica gel as your desiccant. Next, we have your digital balance. So there are two types. It's either the pocket scale or your electronic. So the difference between the two is their capacity and their precision. So for small substances such as those need for weighing in milligrams, so you may actually use the pocket scale. But for greater masses, you use your electronic balance. Next, we have the distilling flask. So as I have mentioned earlier, so this will be part of the distillation setup, and this side arm here is connected to your condenser. So say we have a substance, usually alcohol, and then there is heat being applied, and then there is a thermometer to keep watch of its temperature, and then attached to a cork, and then once evaporation will start, then the vapors will pass through the side arm connected to the condenser and so on. Okay, so it is part of the distillation setup, and this part here is called the neck. So neck. Then this is the side arm. Okay. So next we have here on the right is your dropper. It is familiar, perhaps, medicine dropper, and it has a plastic tube. This part here is detachable. This is your rubber. So even though there is a calibrated markings, you are not to use these when you measure volumes because you'll be provided with graduated cylinder. So droppers are merely for drops of liquid or solution. Okay, so avoid using droppers in measuring liquids. Like for example, you are to measure 5 ml. So don't tell me you're going to use this and then you measure 1 ml, then another 1 ml up to 5 ml. Whereas it's easier if you simply use the graduated cylinder and it is more accurate. Now we have the Erlenmeyer flask, or in other movies or series, or if you do a quick search, it is also pronounced as Erlenmeyer in some parts of the world. But for simplicity's sake, let's call it Erlenmeyer flask. So your Erlenmeyer flask is flat-bottomed. It has a conical body, and then it is usually used for swirling and mixing of liquids without spillage. So the conical shape allows the liquids to simply swirl on its walls, whereas if you use a beaker, it is more prone to spillage outside because the beaker is in cylindrical form, diba? So it is usually used in titration, or even still it's used in heating, but more of the swirling and mixing. So it comes in different sizes, and it is made of glass. Next, we have your evaporating dish. So it is used for evaporation if you have a solution and it has a solute, and then if you have the solid precipitate as well, so you use the evaporating dish to separate the two. So it is made of ceramic and it comes in different sizes also. Next, we have filter paper. So this is a semi-permeable barrier made from cellulose fibers. So you let the liquid pass through, retaining solids, so you separate the two. So for example, you have suspension, so or a colloid, for instance. So liquid will pass through and then the solids will retain on the paper. So we will tackle how to fold this filter paper as soon as we begin with experiment 1. Next, we have the Florence flask. So it is a round body, long neck, and there are two types actually. So you have the flat bottom, or what we call the Florence flask, and there's also what we call the round bottom flask. I have not provided an illustration, but the RBF, or round bottom flask, is still considered a Florence flask. So it is um, used for boiling liquids, and it is also used for sublimation in our one of our experiments. So in sublimation, there will be from gas, there will be solids formed and will adhere to the bottom surface of the flask. Next is the fume hood. So this is a lab equipment. So in our actual laboratory, most of the fume hoods are modified, fabricated. So the main point or the purpose of the fume hood is to protect the user or the one conducting the experiment from the hazardous fumes, especially for concentrated acids and other chemicals. So in some laboratories, there are some with attached gloves here, so you will simply put your hands inside without or with less exposure to the chemicals. But in our lab, since it's just a modified version, so we'll make the most out of it. So next is our funnel. So if you're familiar of, I think in Bisaya it's called embudo. So this funnel, laboratory funnel, is made of glass, but there are also plastic funnels. But for experiments, we use the glass. It is a glass tube or pipe, so you have a conical top and then the narrow tube at the bottom. So it is used together with a filter paper as shown in this illustration, and then the funnel is placed on top of an Erlenmeyer flask. Then in filtration, you pour your solution and then the filtrate will pass through the narrow tube and the residue will retain on the filter paper. Okay? Next, we have your glass or your stirring rod. So it is used to mix liquids or solids in a container, usually in a beaker. So do not confuse stirring rod with thermometer. Okay? Next is your glass tubing. So this is actually a long tube, but it can be cut in pieces or in segments according to usage. Usually, the one who uses the glass tubing the most are pharmacy students. So they have an experiment in the lab which is glass bending or glass manipulation, if I recall correctly. But we can still find glass tubing attached in different setups in our laboratory. So one of which will be distillation setup. So glass tubes are used as connectors. But there are still some like other experiments which utilize glass tubes alone. And it comes in various diameters and it is made of course, glass. The next is the graduated cylinder. So these are hollow glass cylinders with a beak and they are made of glass, and some graduated cylinders have a plastic base, and then some graduated cylinders have a guard or a shield so that if the cylinder falls down it will not break right away. And then graduated cylinder comes in various graduation, So this is used to measure liquids correctly. And we will have another or a more elaborate discussion on how to use graduated cylinders in the succeeding experiments. Next, we have your iron ring. So your iron ring is a metal ring to secure your glassware. So this iron ring will be connected to your iron stand. So it will hold your glassware upright and while it is attached to your iron stand during the experiment. And then your iron stand on the other hand is used to support your other apparatus, usually in heating substances or mixtures using flask or beakers. So those are the typical setups using these two apparatus. Next, we have your litmus paper. So you have two types. You have the blue litmus paper and your red litmus paper. These are small strips that is treated with a natural dye called litmus, and it changes color depending on the pH of the solution. So a little recall. So if you dip your blue litmus paper to a solution or expose it to a vapor and it turns red, meaning that substance or solution is acidic, so BRA. On the other hand, if you dip or expose your red litmus paper to a vapor or a solution and it turns blue, then that solution is or substance is basic. So blue to red acidic, red to blue basic. Next, we have your mortar and your pestle. So these two usually go together. So your mortar is the bowl-like ceramic apparatus and your pestle is the ceramic one that looks like a small baseball bat. So it is used to crush or to pound or to grind substances, especially that in our succeeding experiments we will deal with food and a lot of them will require grounding or diminution into smaller pieces or smaller parts. So these two are really helpful. And mortar and pestle comes in various sizes as well. Next, we have your Petri dish. So Petri dish come in pairs. So you have the lower cover and the top. So this is the bottom cover and the top. Usually it's used for culture, but in our case in chemistry, it is usually used as a container. So usually for solids or it depends on what you use the Petri dish. So it is transparent and made of glass, but originally it's used to culture microorganisms. Next, we have the pipette. So these are small, well, not small, but these are precise volumetric apparatus. So you have their different graduation as well. So there are different types. So if you have 10 ml, you also have 5 ml. So it is used to measure and transfer small amounts of liquids. Okay, so if you notice or if you remember, pipettes are used together with your aspirator or your pipettor. So you are not allowed to use your mouth to suction liquids using a pipette. So that is hazardous and it is dangerous. Okay? Next we have your reagent bottle. So there are different types of reagent bottles. Your reagent bottles are used to store your reagents for safety and organization. So it's either you have your amber bottles or your colorless. So all of these are transparent and the lid or the cap varies. So it's either the narrow mouth type or the screw cap type. But there are also small reagent bottles which may be familiar. So if you notice medicine bottles, so there are also reagent bottles being used like that in the lab. The next apparatus would be your separatory funnel. So it looks like a pear. And then you have here a stop, a valve, and at the top you also have its cap. So in the lab, the cap is plastic or PTFE, same material as this one. Then this tip or edge is similar to that of the edge or the end of the funnel. So, what is the use of the separatory funnel? So, it is used for liquid-liquid extraction. So, you're going to separate the components of a mixture into immiscible solvent phases. So, you will be using this in the experiment later on involving separation of liquids at different phases. Next is your suction flask. So this is basically an Erlenmeyer flask or Erlenmeyer flask with a sidearm, and others call it the Buchner flask, and it has a pair. Usually there's what you call the Buchner or the suction funnel. So the suction flask is basically again an Erlenmeyer, and it is this sidearm is connected with rubber tubing, usually with a pump or a vacuum. Next is the syringe. So syringe are graduated cylindrical barrels with the plunger and then a needle attached to the other end. So this is used to transfer precise volumes of liquids or withdraw liquid samples. So I am certain that you are really familiar because in layman's term, we call this injection. But actually, injection is the process, right, of using one of the processes or one of the ways to use the syringe. And it comes in various sizes. So, 10 cc, 5 cc. So, do note that 1 cc or 1 cubic centimeter is just the same as 1 ml. So, that's a conversion factor. Next, we have test tube. By the way, for safety purposes, remember that when syringe are not in use, you must always put the cap, its plastic cap. And upon disposal, we are going to put this in its designated garbage bin. So, naroon na label sa trash can in the laboratory. Next, we have your test tube. So, these are glass, slender, cylindrical containers. And please let's emphasize that test tubes are not calibrated. They do not have graduation because we do not use them to measure the solution or substances. These are simply used for containing or for testing small scale reaction, okay, or in heating. So if you're going to draw this later on, do not put graduation on the walls of the test tube. So test tubes also come in various sizes. Next, we have your test tube brush. So these are used to clean your test tubes. And then you have your test tube holder to hold your test tubes, especially during heating or carrying exothermic reaction. So exothermic meaning the solution might emit or will release heat during the reaction. Next, we have your test tube rack. So this is usually plastic, but in the lab there is also stainless test tube rack. So it is used to hold, to organize your test tube, just like the ones here at the right side. Next is your tripod. So it is a stand made of lightweight metal to provide stable platform during heating. So I have not yet encountered an actual, but usually clay shield manggod ang ginayuse. So clay shield and the iron ring, and then the maybe sometimes it's the alcohol lamp or sometimes it's the Erlenmeyer flask. So I haven't encountered yet tripod being used. Next is the thermometer. So these are called alcohol thermometers, and before we used to use mercury thermometers. If you're familiar, well, I don't think so familiar pa mo, but if any of you can recall that in measuring or checking out the body temperature, it's the small mercury thermometers being used. But nowadays, digital na due to the environmental laws that prohibit the use of mercury. And we do know that mercury is a hazardous substance. That's why its thermometers are replaced with alcohol as the liquid inside to indicate the temperature. So this still follows the theory or the concept of volume expansion. So alcohol thermometers are still dangerous because first off, if this bulb here will be broken here at the edge, it will give off a really foul smell. And once in contact, maybe the skin might be irritated. Okay? So be careful with using thermometers. Do not use thermometers as a steering rod. I repeat, do not use thermometers as a steering rod. Next, we have the utility clamp. So there are two types. We have the iron clamp and your test tube clamp. So your iron clamp is usually for the condenser in the distillation setup, while your test tube clamp is, well, used for the test tube. Or you may use your test tube clamp in handling or holding funnel, or sometimes you may actually use this clamp for burette in case dili or wala na diri in my use. So as long as the burette will not fall off, then these two clamps are attached to your iron stand. Next is your volumetric flask. So it also looks like a pear. And then your volumetric flask is used for standardization of solutions. So it comes in various sizes, and usually it's being used when you're going to standardize an aliquot of a sample, or an aliquot means a portion of a sample of solution that you're going to titrate later on. The next, we have your water bath. So this is made of copper. It is like a bowl. It has rings around. So over time, frequent usage will lead, just like the burner, color of the burner lead to discoloration. That's why this is now the actual water bath you will see in the lab, but it was once shiny. So water baths are used for heating, but with controlled temperatures, or it may also be used to cool substances. So you're going to put your ice with water or simply water, and then for heating, you're going to fill it with water. Okay, so avoid using the water bath without water because it will lead to damage sa iyang bottom. Next is your wash bottle. So this is made of squeezable plastic. It is used to contain water maintained at a specific temperature. So wash bottles are usually filled with distilled water. Then next we have your watch glass. So this is not contact lens, students. This is your watch glass, and it comes in various diameters. So watch glass is used to used for gentle evaporation of small amounts. So there are two types of apparatus now that you use for evaporation. You have your evaporating dish and your watch glass. But normally, watch glass is used for small amounts only. And it is also used to cover beakers or flask, or simply to watch or observe a certain substance undergoing a reaction. That's why it's called watch glass. Okay? Then last but not the least, you have your wire gauze. So this is very important. This protects your glassware and you as well. So your wire gauze is a flat, mat-like, marsiyag banig, no? Piece of equipment with interwoven metal wires, and there is a ceramic circular center. So do note, the ceramic circular center is not made of asbestos. This is made of ceramic, so modified ceramic. So it is used to prevent overheating and the possible breakage of your glassware. And we're done with the 50 apparatus and equipment. But for engineering, you have one other apparatus not mentioned. That this is your BOD, or your biological oxygen demand bottle. So this is used for collecting wastewater sample, which will be further tested for its BOD. Okay? So that's about it. And instructions. You're going to draw all the apparatus on a long-sized bond paper with 1-inch margin. And you're going to write or caption it with a name and short description. Take note, short description ha. Short description. And you're going to use the cover page, which will be posted on a separate link. So, for questions and clarifications, no need to hesitate to comment, and I hope you learned a lot. See you in the next video.